Dr. Becker here for part 2. We are jumping right into lipids. So lipids are a diverse group of our biological molecules, and they have common features. One main important thing to remember about lipids is their insolubility in water. Fatty acids and their derivatives constitute the main form of lipids and are present in fat and oils. So for fatty acids, we have short, medium, long, and very long chains, which are the building blocks for other lipids. Triglycerides are the main energy storage form in the body, and these consist of 3 fatty acids bonded to a glycerol backbone. We have phospholipids, most abundant molecule in the cell membrane, which consist of glycerol backbone, 2 fatty acid chains, polar head group attached by a phosphate group, and then sphingolipids are fatty acids and polar group attached to amino acid serine rather than the glycerol. They are found in many membranes, particularly in our nervous system. Then also part of our lipids are our steroid cholesterol and compounds derived from cholesterol, which include our steroid hormones and our bile acids. Lipids are hydrophobic and nonpolar. So they don't like water, they are nonpolar, and they cannot travel freely in the body's aqueous solution. So before we go into any more details, let's watch our osmosis just to have a nice review. Remember, osmosis in and of itself is not testable, so if they talk about other terms that we don't talk about, the osmosis stuff is not on the tests or quizzes.
They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a croissant. Fats are also a major component of cells and tissues, and they also help absorb essential vitamins, and can be converted into other molecules like prostaglandins, which help cells communicate with each other. Fats have a 3-carbon backbone called glycerol, as well as fatty acid chains. The fatty acid chain is basically a string of carbon and hydrogen atoms. When an OH group from the glycerol molecule binds to a hydrogen from the fatty acid, an H2O or water molecule gets released, and the two molecules link up. If this happens once, the result is a monoglyceride. If it happens twice, it's a diglyceride, and 3 times makes a triglyceride. Now, there are various types of fatty acid chains, and one way to categorize them is by their length. In other words, how many carbons they have. Short-chain fatty acids have 2 to 5 carbons, medium-chain fatty acids have 6 to 12 carbons, and long-chain fatty acids have 13 or more carbons. Fatty acid chains are also categorized by the bonds connecting the carbons in the chain. A single bond is just one bond between the carbon atoms. And when a fatty acid chain has only single bonds, it's called a saturated fatty acid, because it has as many hydrogen atoms as possible, or it's saturated with them. Triglycerides with saturated fatty acids are nice and straight, so they pack together really well. And as a result, they're usually solid at room temperature. And the longer the saturated fatty acid chain, the more likely it'll be solid at room temperature. Carbons can also have double bonds between them, though. And when a fatty acid has 1 or more double bonds, it's called an unsaturated fatty acid, because it's not saturated with hydrogen atoms. For every double bond, there are 2 fewer hydrogen atoms. Also, a double bond causes a kink in the molecule, so that unsaturated fats don't pack together as nicely as saturated fats. As a result, unsaturated fats are usually liquid at room temperature. Unsaturated fatty acids can be further classified, according to the number of their double bonds. Monounsaturated fatty acids are unsaturated fatty acids with just 1 double bond. Polyunsaturated fatty acids have 2 or more double bonds. Also, they can be classified according to their location as well. Since all these hydrogens can get kind of crazy looking, we'll just take them away for now. So another name for the methyl end is the omega end. And then we can count the number of carbons until the first double bond. Since this one is 3, it would be an omega-3 fatty acid. If the double bond is 6 carbons from the end, it's an omega-6, and if it's 9 carbons from the end, it's called omega-9. Now, to make things even easier when looking at these molecules, I'm just going to show the bonds. Alright, so omega-3s are usually polyunsaturated fatty acids, and include alpha-linolenic acid, or ALA, eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. EPA and DHA are marine sources of omega-3s. They're produced by microalgae and end up in the tissues of fish like anchovies, mackerel, salmon, and sardines. ALA is found in plants like flaxseed, walnuts, and canola and soybean oils. Our bodies can convert ALA into EPA and DHA, but it's an inefficient process that yields only small quantities. And that's why dietary recommendations include foods that have EPA and DHA. Omega-6 fatty acids are also usually polyunsaturated and include linoleic acid and arachidonic acid. Linoleic acid is found in oils like safflower, corn, and soybean oils. Arachidonic acid is found in animal sources like fish, meat, and eggs. Our bodies can convert linoleic acid into arachidonic acid, but once again the process is inefficient. Because ALA and linoleic acid can only be obtained in the diet, they're considered essential fatty acids. Omega-9 fatty acids are typically monounsaturated fatty acids, and an example would be oleic acid, and these can be made by the human body. Foods like canola and olive oil, as well as almonds, contain omega-9s. Now, looking at the double bond of this unsaturated fatty acid, like most unsaturated fats, it's got a cis configuration. In a cis configuration, the two functional groups are on the same side of the double bonded carbons. Now, when this happens, the fatty acid chain naturally bends. A molecule that bends does not pack tightly together, so it's a lot more fluid. Think about cooking oils, which are liquid at room temperature. Some fats are in a trans configuration, though. In a trans configuration, the functional groups are on opposite sides of the double bonded carbons, and this keeps the chain more straight and easier to pack. Trans fats result from a process called partial hydrogenation. In just plain old hydrogenation, hydrogens are added to cis fats to get rid of all of the double bonds. So let's say that you've got this triglyceride, and it has a total of 2 double bonds. So in this case, we'd add 4 hydrogens, 2 for each double bond. That turns the unsaturated fatty acids with cis double bonds into saturated fatty acids. Partial hydrogenation, on the other hand, refers to adding hydrogens to most, but not all double bonds. So let's say now that we add just 2 hydrogens. When this happens, some double bonds can be turned into single bonds by the hydrogens, but then they might reform. And what you end up with is an unsaturated fat, but some of those fats have trans double bonds. Partial hydrogenation is a process that happens naturally in the digestive tract of some animals, like cows and pigs, which is why trans fats can be found naturally in meat and dairy products. Partial hydrogenation is also sometimes used in the food industry, in the processing of liquid oils to make them more solid, which can actually lead to the creation of trans fats. Partially hydrogenated oils have been largely removed from foods in North America and Europe, because trans fats have been associated with coronary heart disease. Although some foods might have more of one type of fat than another, the truth is that all foods are made up of a blend of fatty acids. When you eat a food like peanut butter, which has about 75% of its calories from fat, the body goes through a set of steps to digest and absorb the fatty acids. First of all, triglycerides are hydrophobic, so they form large globules of fat, like what you see when you pour oil in water. Enzymes called lipases in the saliva, stomach, and secreted by the pancreas can break down triglycerides into free fatty acids and monoglycerides. But working on the surface of a globule is inefficient. So to speed things up, bile salts produced by the liver break the large fat droplet into smaller droplets, which increases the surface area for the lipases to work. Once the triglycerides are broken down into monoglycerides and free fatty acids, these self-assemble into mixed micelles, which have a hydrophobic interior and a hydrophilic or water-loving exterior. The micelles glide through the watery environment of the intestinal lumen and reach the enterocytes in the intestinal wall. When they get to the enterocytes, the micelles release the fatty acids and monoglycerides, which diffuse into the enterocyte. Inside the enterocyte, the fatty acids and monoglycerides reassemble into triglycerides, and these get packed into a larger structure called a chylomicron. The chylomicron has lipids and proteins, so it's a lipoprotein. It has an outer membrane with phospholipids and proteins, and a hydrophobic core that has triglycerides, cholesterol, and fat-soluble vitamins A, D, E, and K. The chylomicron then leaves the enterocyte, but it's too large to get into the endothelial cells, so instead it enters a nearby lymphatic capillary called a lacteal. From there, the chylomicron floats in the lymph and flows into the thoracic duct, and then gets dumped into the blood, essentially bypassing the portal vein. Once in the blood, the chylomicron releases fatty acids and monoglycerides in peripheral tissues like muscle, which use them for energy, as well as adipose tissue which can store them. After delivering the triglycerides, the chylomicron shrinks in size and eventually gets engulfed by the liver. Now, fats play a super important role throughout the body. They have a huge number of health benefits, and those benefits can vary by the type of fat we eat. For example, polyunsaturated fats are precursors for hormone-like molecules called prostaglandins that stimulate endothelial cells that line blood vessels to release nitric oxide. Nitric oxide is a vasodilator, so that decreases resistance to blood flow and in turn lowers blood pressure. Polyunsaturated fatty acids can also help reduce the total and LDL cholesterol, and that's linked to lower rates of cardiovascular diseases like heart attacks and stroke. Long-chain omega-3 fatty acids like DHA and EPA can both help to lower plasma triglyceride levels, which also protects against cardiovascular disease. Finally, DHA is important in the development of eyes and brains of young infants. The impact of saturated fat on cardiovascular health is a little more complex. Generally speaking, it's recommended to keep consumption of saturated fat low, but just like with unsaturated fats, there are different types of saturated fats. Evidence suggests that different types of saturated fatty acids might have different effects on our cardiovascular health. Evidence also suggests that the health impact of reducing or replacing saturated fat in the diet depends on the nutrient that replaces it. For example, replacing saturated fat with polyunsaturated fat has been shown to benefit cardiovascular health, whereas replacing saturated fat with refined carbohydrate has not. Based on this, the National Academies of Medicine recommends consuming 20 to 35% of our daily calories as fat for a 2,000 daily calorie intake. That comes out to between 400 and 700 calories from fat, or about 44 to 78 grams. And it's not just about the amount of fat that we eat. The type also matters.
So this here was covered, the idea of how these look. The triglycerides is synthesized by a dehydration reaction, adding 3 fatty acid chains to a glycerol molecule. This one here is showing the phospholipids, saturated fatty acid, and a monosaturated fatty acid, and the difference of how they look structurally. Also goes into cholesterol. So to continue to kind of review on these, let's talk about the different structures and functions then a little bit more individually. When we talked about the fatty acids, kind of go through, there it is. So the fatty acids are transported in the blood, bound non-covalently to the serum protein albumin. Fatty acids have different configurations based on the bonds between the carbon atoms and the hydrocarbon chains. We talked about saturated fatty acids, which would be here. Those are carbon atoms are saturated with as many hydrogen atoms as possible, thus making only single bonds with adjacent carbons. This is showing stearate, a saturated fatty acid that contains 18 carbons. And in this figure, the carbons are omitted for simplicity, and each straight line represents a single bond between the 2 carbons. The unsaturated fatty acids have a fewer hydrogen atoms because they contain at least 1 carbon-to-carbon double bond, one with the next carbon in the chain and the other with a hydrogen. So in unsaturated fatty acids, not all the carbons are saturated with 2 hydrogen atoms, so they're not saturated. The carbons that form a double bond may assume 2 possible configurations. The double, which is one of them, is the trans fatty acid. The double bond within the fatty acid is in cis configuration when the hydrogens bound to adjacent carbons are on the same side of the molecule. And trans fatty acids configuration is when the bound hydrogens are on the opposite side of the molecule. Which is this one here. It's important that we remember that biological membranes contain both saturated and unsaturated fatty acids. And when the saturated fatty acids predominate, remember there's a lot more hydrogen, they can be really tightly packed, which increases the rigidity of the membrane. However, because unsaturated fatty acids have that kink in the molecule, they tend to occupy more space, leading to a membrane that has increased fluidity and flexibility. So the example that the book gives, I think, is really good. And it says, consider butter and oil. The former, which is butter, is a form of animal origin, and roughly 60% of its fatty acids are saturated and 40% are unsaturated. Like oil is plant origin, and 80% of its fatty acids are unsaturated. So at room temperature, the fatty acids in butter are tightly packed, resulting in a solid substance. So they're saturated fatty acid. At room temperature, the unsaturated fatty acids in the oil confer fluidity and make the substance liquid. Since unsaturated fatty acids are subject to oxidation and can become rancid, the process of artificial partial hydrogenation was developed to lengthen the shelf life of oils. So they converted some of this double bonds, the cis double bonds, into single bonds, and some of the cis double bonds into trans double bonds. And that's where we get formation of partially hydrogenated oils. Unfortunately, we now have pretty compelling evidence that dietary intake of trans fatty acids, or trans fat, will increase the risk of developing cardiovascular disease because it's going to raise the level of the so-called bad cholesterol, which is our LDL. So in light of those findings, the FDA established that the partial hydrogenated oils, or PHOs, which are the primary dietary source of industrial produced trans fatty acids, are not recognized as safe and are considering banning them. There is naturally occurring partially hydrogenated oils that are allowed to be present in food, but we're not allowed to artificially add them. Then they started talking about the differences of the cholesterides. Triglycerides are the major form of energy storage, as the hydrocarbon chains can undergo catabolic reactions that will release that large amount of energy. Triglycerides are mainly stored in our fat tissue, adipose tissue, where lipase enzymes catalyze the reaction that will release fatty acids and glycerol so that they can be exported to tissue and organs that need energy. Then we go into the eicosanoids. So certain lipid components of our biological membranes, such as arachidonic acid, may be released and converted into paracrine hormones. These are substances that act near their production site. So arachidonic acid is a 20-carbon unsaturated fatty acid containing 4 cis double bonds that give it a hairpin shape. This is that right here. It is the precursor for many lipid molecules that participate in inflammation and other homeostatic responses, such as thromboxane, prostaglandins, and leukotrienes, which is what we have here. Those molecules are collectively called eicosanoids, these here. And it's actually from the Greek word for 20. Eicosanoids are involved in the physiologic process for inflammation, which control of blood flow and pressure and reproductive functions. So understanding their structure and their synthesis led to the development of our NSAIDs. Woohoo! Specifically, the drugs that can inhibit the cyclooxygenase enzymes, which catalyze an early step in the conversion of arachidonate to prostaglandins and thromboxane. And this mechanism will allow aspirin and ibuprofen to decrease pain, fever, and the inflammatory response in general. And we made our COX inhibitors based on our understanding of the eicosanoids and how they lead to inflammation. So when we have anti-inflammatory medications, we are manipulating the arachidonic acid pathway. We can inhibit the COX, or we can inhibit the 5 LOX and the NSAIDs. We'll talk more in Chapter 4 about some of those other topics to talk about. So cholesterol and its derivatives is important to understand. And I'm not going to get into too much of that, but we talked a little bit before about phospholipids are the main lipids that make up the cell membrane. Cholesterol is inserted in the smaller amounts and is important at stabilizing the membrane fluidity for normal function of the membrane. Cholesterol is also a precursor for all steroid hormones, androgens, estrogens, progestins, glucocorticoids, and mineralocorticoids. These hormones regulate various functions in the body, ranging from the development of our sexual characteristics all the way to electrolyte imbalances. All of the hormones are hydrophobic in nature, which makes sense if they have cholesterol, their derivative. So their mode of action is fundamentally different from that of protein hormones, and we'll talk more about that. Next, we get into lipoproteins. So the lipoproteins are essentially the lipid packages for plasma transport. So the triglycerides and cholesterol are insoluble, so they do not like water. The lipids are packaged in lipoprotein particles coated with phospholipids and proteins. The proteins target particles to their specific uptake sites. Okay. So as the name applies with the lipoproteins, these substances have a core that are made of lipids, but then the outer core is made up of phospholipids in a form of protein called apolipoproteins. So apolipoproteins have a twofold role. One, they coat and solubilize the lipids, and they regulate lipid exchange between blood and tissues. So based on their composition and associated density, role, and origin, lipoproteins are classified into several different categories. We have chylomicrons, VLDLs, intermediate density lipoprotein, LDLs, and high density lipoproteins. And if you go into the table 2.2 in the chapter, you can see those there, where their sources, and kind of if they're cholesterol or triglycerides or et cetera, and what the name of the apolipoproteins are. So the chylomicrons are made by small intestine cells from dietary fats, and they're the last step in digestion and absorption of dietary fats. So they enter the circulatory from the lymphatic circulation, and then they acquire other apolipoproteins. The chylomicrons consist mostly of triglycerides, but they also contain phosphatidylglycosine, cholesterol, and very little protein. They're very high fat content. Their role is to deliver triglycerides to the tissues in the body, particularly to muscle and fat. So they carry lipids from the dietary source to the muscle and fat tissues. So this pathway of lipid delivery is referred to as the exogenous pathway. Once these particles have released the majority of their triglyceride content, what remains is called the chylomicron remnants, and then it's cleared by the liver. The very low density lipoproteins are made of triglycerides synthesized in the liver. VLDL carries lipids produced by the liver, and that's an endogenous pathway. The particles that are left after VLDLs release their triglycerides to the peripheral cells are called IDLs. The liver is responsible for clearing some of the IDL particles, while the rest are converted to LDL by hepatic lipase. This process is very important to understand when you get into the cardiovascular system and when you're talking about cholesterol levels and how to manage high cholesterol and high triglycerides, because it's within this system that we've developed our medications to disrupt these pathways. So because the liver is responsible for clearing some of the IDL particles, but others are converted by hepatic lipase, LDL particles are most commonly made of cholesterol, and their main purpose is to release cholesterol to cells throughout the body before being cleared by the liver. So individuals who have high dietary intake of cholesterol and saturated fat will accumulate excessive amounts of LDLs in their circulation, which eventually will migrate into arterial walls, promoting atherogenesis and the development of atherosclerosis, which is bad, right? That's how we're going to get our heart attacks. The HDLs are made by the intestine and liver and circulate cholesterol from tissues back to the liver. They have the highest density of all the lipoproteins because they contain a large amount of protein. They also contain the highest phospholipid content as well as some cholesterol, but very little triglyceride. The HDL particles are synthesized in the liver and intestine to be released into circulation, where they are responsible for transferring apolipoproteins to nascent chylomicrons and VLDL. So low HDL levels are markers of increased risk. This is a lovely little image here that I think it's worth talking about. This is figure 2.0 here. So we can see we have dietary fat and cholesterol that we absorb through our intestines. Through the lymph, we release the chylomicrons, which go into the capillaries. They release what they need to release, and the remnant cyclomicrons come out, and they also release their cholesterol. That cholesterol will go into the liver to be synthesized. It also goes on through our capillaries, and it hydrolyzes lipoprotein lipase, hydrolyzes triglycerides in the fat and muscle. It moves on to here where we convert into VLDL, and some of that continues to go through into IDL, or it gets deposited in the fat and muscles. The IDL, some of it goes to the liver for biosynthesis of fats and cholesterols. Or the IDL here through the hepatic lipase converts into LDL. Some of that goes to the liver, some of that works with the LDL receptor to form cholesterol, and this can go into the peripheral tissues, muscles, and organs. There's also this part here, which then breaks down part of that and goes back out into the IDL. And this cycle here is something that our drugs we have will interrupt. Also, the liver will release pre-HDL out into here, and then our HDL are synthesized in the liver and the intestine to be released into circulation, where they're responsible for transferring apolipoprotein to nascent chylomicrons and VLDL, collecting cholesterol from this LDL and IDL and getting the remnants and then carrying it back to be gotten rid of. So the HDL is very important to have to work. So this shows here all these highlighted in blue had a hydrophilic layer, so proteins, phospholipids, etc., and they aren't going to dissolve in water. Whenever you have a mutation in apolipoproteins, they can lead to dyslipidemia. So you can also have genetic things that can happen to cause high cholesterol genetically. So familial chylomicronemia syndrome is also a name for that. All right, then we talk about amino acids for building blocks and being the building blocks of protein. The proteins perform an exceptional variety of functions in the body. So we're going to jump into those proteins. And amino acids have a central carbon bonded to a hydrogen, an amino group, a carboxylic group, and a variable group, usually abbreviated as the R. There are 20 unique amino acids, and the properties of the variable side chains are used to categorize the amino acids into different groups. So because the amino acids are used to build protein, their side chains extend away from the peptide backbone and determine folding properties and binding sites based on their group characteristics. So we need to watch the osmosis on the proteins. They do a great job as well of explaining this.
Fats are an essential part of a healthy diet. They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a human diet. It's found in a variety of foods like eggs, dairy, seafood, legumes, meats, nuts, and seeds. Regardless of the source, the protein that we eat gets broken down and reformed into new proteins in our bodies. These proteins do everything from fighting infections to helping cells divide. You name it, they're doing it. At its simplest, a protein is a chain of amino acids bound to one another by peptide bonds, like a string of beads. These strings get twisted and folded into a final protein shape. When we eat protein, it gets broken down into its individual amino acids. Most amino acids have a central carbon atom bonded to one amino or nitrogen-containing group and one carboxylic acid group. That's why it's called an amino acid. The carbon also has one hydrogen atom and a side chain, which is unique to each amino acid. The exception to this is proline, which has a tiny little ring structure instead. Although there are hundreds of amino acids in nature, humans only use about 20 of them to make basically every type of protein. They include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. That's 20. One way to divide them is by defining which ones our bodies can make and which ones we can't. There are 5 amino acids, alanine, asparagine, aspartic acid, glutamic acid, and serine, that we can get from foods, but we can also make ourselves. These are called non-essential amino acids. Then there are 6 that we call conditionally essential because healthy bodies can make them under normal circumstances: arginine, cysteine, glutamine, glycine, proline, and tyrosine.
but we can't make them in cases like starvation or certain inborn errors of metabolism. Finally, there are 9 of them that we can only get from food: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. We call these the essential amino acids. Dietary protein provides the essential amino acids that are needed to make our own proteins, hormones, and other important molecules, a circle of life of sorts. But to do so, we need to break the dietary protein down first, through a process called proteolysis. When we first eat a protein-containing food, proteolysis begins when the food reaches the stomach. First, hydrochloric acid denatures the protein, unfolding it and making the amino acid chain more accessible to enzymatic action. Then, pepsin, which is a protein itself made by gastric chief cells, enters the picture. Pepsin cleaves any available protein into smaller oligopeptide chains, which move into the duodenum, where a second set of digestive enzymes, made by the pancreas, further chop the oligopeptides into tripeptides, dipeptides, and individual amino acids. These can all be taken up to the intestinal cells, where di and tripeptides are then converted into amino acids. Some amino acids remain in these cells and are used to synthesize intestinal enzymes and new cells, but most enter the bloodstream and are transported to other parts of the body. In general, animal-based protein foods like eggs, dairy, seafood, and meat provide all 9 essential amino acids in adequate amounts. Soy foods are unique in that they are plant-based and also provide all 9 essential amino acids in adequate amounts. Most other plant foods, including whole grains, legumes, nuts, and seeds, have high amounts of some amino acids and low amounts of others. Hearing this, it might be easy to assume that animal-based foods provide more protein than plant-based ones. But, as it turns out, a cup of tofu has the same number of grams of protein as 3 ounces of steak, chicken, or fish. And half a cup of lentils has more grams of protein than an egg. And not all plant foods are low in the same amino acids, so eating a variety of plant-based foods can provide all 9 of the essentials. For example, pairing protein sources, like rice and beans, or hummus and pita bread, or oatmeal topped with almond butter. However, in terms of volume, it may be necessary to eat more plant-based foods to get a similar amount of protein and amino acid profile provided by animal-based proteins. Generally speaking, daily protein requirements are based on studies that estimate the minimum amount of protein needed to avoid a progressive nitrogen loss. The World Health Organization guidelines and the US recommended dietary allowance each estimate that daily protein requirements for healthy adults are about 0.8 grams per kilogram of body weight. Protein recommendations per day vary by age as well. Children 1 to 3 years of age are recommended to get 13 grams. For ages 4 to 8, 19 grams are recommended. And between ages 9 and 13, 34 grams are recommended. Whether a person is male or female also impacts protein needs. Females ages 14 and above are recommended to get 46 grams of protein per day. Males age 14 to 18 need slightly more, about 52 grams per day. And males 19 and older are recommended to get 56 grams per day. Some groups, like pregnant and breastfeeding women, as well as athletes, have elevated needs, and older adults may also benefit from eating more protein. Getting the right amount of protein per day can be achieved in multiple ways. For example, 1 serving of Greek yogurt with breakfast, a salad topped with 3 ounces of chicken for lunch, and 3 to 4 ounces of fish at dinner provides about 64 grams of protein. Getting the same amount of protein could also be achieved by eating a cup of tofu scramble and a slice of peanut butter toast for breakfast, a cup of shelled edamame with lunch, and 1 cup of lentils and brown rice at dinner, which also offers about 64 grams of protein in total. Now, the fact is that it's still unclear what an optimal amount of protein is, and the research is ongoing. In clinical settings, there are certain individuals who may be at risk for a protein deficiency, including patients with malnutrition, trauma, and burn injuries, as well as various conditions impacting nutrient absorption like inflammatory bowel disease. These individuals may have increased protein needs compared to the general population. Except for certain circumstances like kidney disease, there usually isn't a health risk associated with eating a lot of protein, because our bodies are able to process it. Alright, as a quick recap. Protein is a cornerstone of the human diet, and a major component of our bodies. There are 5 non-essential, 6 conditionally essential, and 9 essential amino acids. We need to get essential amino acids from our diet. Protein needs vary depending on life cycle stage, level of physical activity, and health status. Some health conditions may put people at risk for protein deficiency or increased protein needs. Everyone, whether omnivorous, vegetarian, or vegan, can get enough protein by eating a variety of foods.
Good recap of amino acids and proteins for a good review for us to kind of launch from. Get to my right slide here. All right, so next they talk about the nonpolar hydrophobic amino acids. So the amino acid classes and their properties are different between each of the different functional groups. So the proteins perform an exceptional variety of functions, right? And they can form enzymes, they can support structures, make hormones, etc. So the way that they are sequenced makes them unique. So the amino acids are the building blocks of the proteins. The backbone of all amino acids is characterized by a central carbon, and that's the alpha carbon, typically, which is bound to an amino group, a carboxylic group, a hydrogen atom, and a unique side chain, the R group, which is what we can see in these examples here. The fact that every amino acid has a carboxyl group and an amino group means that individual amino acids are generally polar and hydrophilic, right? So they have that electronegativity gradient that are going to make the cells lean or gravitate more towards one side, making them polar. And polar cells are more hydrophilic. They like water. They also contribute to our process of our pH buffering. And in general, also, each R group has distinctive chemical characteristics that helps determine a protein shape and therefore its biological functions. So this next couple of slides here is going to show the amino acids grouped into the following types based on their R group structures. So this one here is nonpolar hydrophobic amino acid group. Okay, so glycine, alanine, valine, proline, phenylene, tryptophan, methionine, isoleucine, and leucine. This one here are uncharged polar amino acids, glutamine, asparagine, cysteine, tyrosine, threonine, and serine. This one is the charged amino acids. And they're, let me see here and make sure I've got the right one. Those are the uncharged. I think we're missing a slide. Maybe perhaps. Nope, these are acidic right here. Glutamine, aspartate. Yep, we're on the right slide. So these are negatively charged acidic. And then these ones over here are the positively charged basic. And then there's another group called hydroxylic that we don't have on here for the slide. So these are the nonpolar hydrophobic amino acids. So they are not polar, so they don't like water. They don't completely dissolve in the water solute. The simplest amino acid is glycine, which has a hydrogen atom as its R group. Because of its structure, the R group in glycine does not contribute significantly to the development of hydrophobic interactions. Thus, glycine is often classified separately from the other nonpolar amino acids. Alanine is another simple amino acid, as it has a methyl group as a side chain. Three slightly larger amino acids are the valine, isoleucine, and leucine. Although the hydrocarbon chain in valine has 3 carbons, isoleucine and leucine are both made of 4 carbons that are arranged in different ways. These chains confer hydrophobicity to the molecule. Methionine contains a sulfur group that is slightly more polarizable than hydrocarbon chains, yet it is predominantly hydrophobic, and thus often considered part of the nonpolar amino acids. Then you have your tryptophan and your phenylalanine, and these are also called aromatic amino acids because they contain aromatic rings. Tryptophan is made of 2 ring structures that contain a single nitrogen. So in essence, if someone has asked you, give me examples of nonpolar hydrophobic amino acids, you can pick from these on the screen here. The uncharged polar amino acids is the second class here. This is cysteine, asparagine, and glutamine. These ones right here. Cysteine was introduced earlier in our discussions up top of the chapter here because it contains a thiol group that allows it to form disulfide bonds, and it will contribute significantly to the development of 3-dimensional structures in proteins like insulin. Then we have asparagine and cysteine, which are also polar structures that make them hydrophilic. Then we have our acidic amino acids and the basic amino acids. This one here is the hydroxylic amino acids, which are uncharged polar amino acids with a hydroxyl group. These are polar and uncharged, yet they are discussed separately to emphasize their special role in cell signaling. So, we talked about the kinases are enzymes that can transfer a phosphate group from a molecule, usually like ATP, to an acceptor. When the acceptor is a protein, phosphorylation usually takes place on hydroxyl groups of serine, threonine, and sometimes tyrosine. So the hydrogen of the hydroxyl is displaced by the phosphate group in that reaction. So common signaling cascade in cells, like those that are activated by our neurotransmitters or by our hormones, often involve serine threonine kinases, and it's that modification of the protein activity by phosphorylation that's the common pathway of cell adaptation to maintain homeostasis. So, in other words, it is this group of the uncharged polar amino acids. Because they have a hydroxyl group, we refer to them as hydroxylic. And the two which are involved, serine and threonine kinases, often work in phosphorylation. And the tyrosine kinases will contribute to cell signaling by growth factors, cytokines, and hormone insulin. And it's the tyrosine that can be implicated in neoplastic transformations in cancer. So there's a drug called a TKI, which we're going to learn about in pharmacology, which is a tyrosine kinase inhibitor that's going to block this amino acid to help prevent the proliferation of cancer cells. So this is a very special group of amino acids that further down the line you're going to hear of again. So again, we typically say that the amino acids are building blocks, right, for protein synthesis, but these individual amino acids also can serve very specific functions in the body. Remember that our individual amino acids can be used as energy sources because they can be used as substrates for gluconeogenesis during fasting, right? So we know that after you burn through all of your fat, you will begin to break down muscle, right? And use that for energy. So that protein is going to be broken down through gluconeogenesis and created changed into energy for our body during fasting or like starvation, right? Also, tyrosine, like we talked about, is a precursor for our melanin. It's needed to transmit dopamine and norepinephrine, and we have to have adequate tyrosine for our thyroid hormones, which is all deficit in patients with phenylketonuria, PKU, which is what is screened for at birth. And it's mandatory. All children in America, quote unquote, have the right to be screened for PKU because it can be devastating and it's fixable if it's found. Tryptophan is the precursors to serotonin and melatonin, which is why we think when you eat turkey, it's the tryptophan that triggers the melatonin and makes you sleepy. Histidine is the precursor to histamine, which is an inflammatory signal and a neurotransmitter. Glutamate is the excitatory neurotransmitter and the precursor of GABA. So when we talk about glutamate again in pharmacology, this is where that plays in as well. Glycine is the precursor for heme, which is necessary for us to make hemoglobin and myoglobin. And then we have hydroxyl-containing amino acids, again, are the target of second messengers in hormone signaling. So yes, all of the different amino acids can be folded together to make protein and different building blocks for protein, but in and of themselves, they can have also specific functions to themselves. So when we talk about how do we synthesize or create and make protein from our amino acids, proteins are linear polymers of amino acids linked together by peptide bonds in a dehydration reactivation. So proteins have an amino end, which is a leading edge during synthesis, and a carboxyl end, which is the end of the protein chain, which you can see here has the carbon, carboxyl. When the carboxylic group of one amino acid is bound to the amino group of the neighboring amino acid, a hydroxyl group and a proton are released in the form of water in dehydration reaction. When you have two amino acids bound together by a peptide bond, they are called a dipeptide. And the term peptide typically refers to a short chain of less than 30 amino acids. Peptide bond formation takes place during protein synthesis. Like they talked about in the osmosis video, we have to discuss protein folding, okay? Because during protein synthesis, these amino acid sequences favor the formation of secondary structures, and how the structures lay and fold ultimately determine how the protein is made. The two common motifs that are stable secondary structures of peptide chains are the alpha helix and the beta sheet. In an alpha helix, the peptide is coiled to form a single right-handed helix in which the hydrogen bonds run parallel to the main axis of the helix here. The carbonyl groups point in one direction of the helix while the amino groups point in the opposite direction. Another very common secondary motif is the beta sheet or beta pleated sheets. In this case, the hydrogen bonds are formed between different sequences of amino acids that lie parallel to each other to form a sheet. So kind of like stacking sheets of paper on top of each other. And the amino acid sequences may be oriented in the same direction, giving rise to a parallel configuration, or in the opposite direction. In both cases, the hydrogen bonds remain on the same plane, right? So this one, they're all in the same direction. On this one, they're opposite directions, but they all stay on the same plane. Then we can have secondary structures of the large proteins then are further assembled to form a tertiary structure. So if you look in your book on figure 2.22d, this shows the representation of the protein angiogenin. Here, the beta sheets are represented by the blue arrows here. The alpha helices are in the red here. The process of folding and then consolidating the alpha helices and the beta sheets of the protein and their connecting loops is a critical event for proper function. This process is assisted by chaperone proteins in the endoplasmic reticulum that manage the assembly of the final protein into the proper form, and they eliminate incorrect folding. Proper folding is such an important requirement for these complex structures that the endoplasmic reticulum has a detection system for improperly folded proteins. The unfolded protein response, which is the UPR, is capable of stopping all protein production by cells and either restoring the normal function or inducing apoptosis, which is programmed cell death, when a protein misfolding is detected. That's pretty cool. Much of the folding that occurs in formation of the tertiary structure of the protein is stabilized between the interactions of the amino acid side chains, which are the R groups. A key determinant, though, is the hydrophobic interactions between the nonpolar R groups. So protein folding of intracellular and secreted globular protein functions to position the hydrophobic region in the inner core of the protein, while exposing the hydrophilic regions as a coat facing the water environment of the intracellular fluids. This is the strategy known as stabilization by hydrophobic interactions, which is this one here. So proteins that are destined to remain in the intracellular fluid or to be secreted to the plasma must be compatible with water environment, right? We said that most of these have to function within water because that's our main transport system. So they are synthesized or folded in a specific way and shape that will display the water-loving ends on the outside of the protein with the water-hating ends clustered in the inside. So this here, the yellow is water-loving and the pink is water-hating. So instead of having this protein float through the water like this, which would not work very well, it's going to be folded into this specific structure so that the water-loving parts face the outside and it can travel through the water, so to speak, better. It'll interact with the water better. Then we talked, or the book goes into the quaternary structures. Quaternary, yeah. Quaternary, I'm not really sure. Quaternary structures, I'll say that. The quaternary structures, either one. Yeah, I don't think I'll probably say it the same way twice. Anyway, the structure of the proteins is complete at the tertiary level, right? And those proteins are called monomeric. But other proteins then have an additional level of the organization, the quaternary structure. In these cases, the functional unit is made of two or more proteins, leading to the formation of a multimeric protein. The example is dimers, trimers, and tetramers are made by assembling 2, 3, or 4 protein subunits, respectively. So the example is oxygen-carrying protein hemoglobin, which is a tetrameric protein as it's made of 4 subunits. So each subunit consists of globin, protein, and the heme. The heme is represented in gray here, and it is a form of porphyrin that contains an iron atom in its center that is responsible for oxygen binding, right? And then there are the 2 types of globin subunits, hemoglobin alpha and beta. And that is the blue and the red. Because there are 4 subunits, each hemoglobin molecule can bind up to 4 oxygen molecules. So oxygen carrying capacity of hemoglobin is a quaternary structure of proteins with multiple subunits. Protein will perform in a very varied number of functions within our body, and I wanted to talk about that a little bit more in detail. So like, what do the proteins do? What function do they serve? 1, they allow movement. 2, they provide structural support. They work as hormones, enzymes, transporters, and more. So, proteins like myosin are able to convert the chemical energy stored in ATP into mechanical energy that's then used for many types of cell and body movements. The proteins, some proteins are structural components that shape and support tissues and organs, like the ground substance of connective tissue that is made of proteoglycans, which are proteins bound to carbohydrate residues. Other proteins will confer unique properties to the tissues. An example of that is elastin will confer elasticity to the walls of the alveoli in the lungs, allowing for elastic recoil that drives our expiration. Collagen confers tensile strength to our bones and our connective tissue. Without collagen, right, the bones would be made of brittle crystals that wouldn't work very well. So proteins have an extraordinarily wide array of functions within the body, and they're found in all parts of the body. They're absolutely one of the top 4 things that we need for the human body, for sure. It's also important to realize that protein hormones are synthesized by endocrine glands, that they can be released into the circulation, traveling to the target cells. Membrane receptors activate multi-step signal transduction inside cells, and those pathways allow the cells to finally regulate, amplify, and activate the downstream targets. We have to have plasma proteins to bind or transport the hydrophobic compounds, right? So we know there's things that need to be transported throughout the body that don't like water. They're hydrophobic, and we need our proteins to be the transport to carry the hydrophobic compounds to the parts of the body that are needed, or we need it to carry substances with high affinity, such as hemoglobin. It's a protein that carries our oxygen. So those are some of the examples to really highlight the different functions that the proteins have. Other proteins then are transmembrane transporters. So they will allow water-soluble molecules across biological membranes. The ion channels and the ion pumps are also integral proteins that allow the movement then of ions from one side of a membrane to the other. So we need the proteins for that as well. All right, next we move into nucleotides. Nucleic acids, such as our DNA and our RNA, are linear polymers of nucleotides, which in turn are made of nitrogenous base, a pentose sugar, and one or more phosphate groups. So we have those small molecules that polymerize to make up nucleic acids, which are our DNA and our RNA. There's the nitrogenous bases, which include the pyrimidine and the purine bases. Pyrimidine consists of a 6-membered aromatic ring that contains 2 atoms of nitrogen. Purines are made a little differently. The 3 pyridamine bases are the C, U, and T, right? So cytosine, uracil, and thymine cut are, I always remembered this as cut the pyramid. So pyrimidine bases are cut, cytosine, uracil, and thymine. The uracil is in the RNA. And the 2 purine bases are AG, adenine and guanine. And then we have the nucleotide trisephosphates, which are like ATP, CTP, GTP, TTP, and UTP. Those are joined together at the site of the phosphate attached to the 5th carbon of the sugar and the hydroxyl attached to the 3rd carbon of the sugar. So this is referred to as synthesis from 5' to 3' direction. All right, ATP is the primary energy currency in the body. And that means it's the energy for all cells. So the phosphate groups are bound by high energy bonds. So the release of the phosphoryl group from ATP is used to supply energy for many cellular functions and enzyme reactions. Okay. With 3 high-energy phosphate bonds that can be used to power enzyme reactions, membrane transport, muscle construction, and other cell activities. ATP is a special major energy source for our cells. The enzyme adenylyl cyclase converts ATP to cyclic adenosine monophosphate, which we call CAMP, in response to stimulation by hormones and transmitters. So CAMP is an intracellular second messenger that alters enzyme activity in accordance with homeostatic needs. GTP is our guanosine triphosphate, and that can be converted to the second messenger signal cyclic guanosine monophosphate, which is cGMP. So guanosine triphosphate, GTP, is also used as a timing mechanism in membrane receptor signaling cascades. So let's pause here before we go on and listen to another osmosis.
Amino acids are the building blocks of proteins. And just like how you can make lots of words with a finite alphabet, it's possible to make lots of proteins with just 20 amino acids. Each amino acid has a nitrogen-containing amine group and a carboxylic acid, hence the name amino acid. Each amino acid also has a unique side chain that's kind of like the amino acid's fingerprint. 9 of the 20 amino acids are essential, meaning that you obtain them from dietary sources rich in protein, like meats or tofu. The other 11 amino acids are non-essential, which means that they can be made in our body, so you do not have to get them from your diet. So let's say you had a nice big bowl of lentils, rich in protein. Protein would get broken down into amino acids, and those amino acids would make their way into various cells to serve as building blocks in protein synthesis. And the cell has to try to make use of these amino acids, because ammonia, which is the nitrogen-containing amine group in amino acids, can become toxic to the cell if it gets freed up and starts to build up in the cell. Ultimately, to get rid of it, ammonia must first be removed from the amino acid and then sent to the liver where it can get metabolized into a less toxic molecule called urea. To do that, a group of enzymes called transaminases or aminotransferases transfer that nitrogen-containing amino group from amino acids to keto acids, like alpha-ketoglutarate. These reactions are called transamination reactions, and they're reversible reactions, meaning that the reaction can go in either direction using the same enzyme. And generally speaking, transamination reactions require pyridoxine, or vitamin B6, as a cofactor to help move things along. So let's take an example of a transamination reaction with the amino acid alanine in a muscle cell. First, the enzyme alanine transaminase, or ALT, switches the amino group on alanine with the oxygen group on alpha-ketoglutarate, resulting in a keto acid called pyruvate, which now has the oxygen group, and the amino acid glutamate, which now has the amino group. Now pyruvate has two options. First, it can be converted into acetyl-CoA by pyruvate dehydrogenase in the muscle cell, and then the acetyl-CoA can enter the citric acid cycle. Second, pyruvate can be converted to lactate by lactate dehydrogenase, and lactate can then travel to the liver. Because this is a reversible reaction, lactate can be reconverted back to pyruvate in the liver by lactate dehydrogenase as well. Within the liver, pyruvate can enter gluconeogenesis and help form a new glucose molecule. Glucose can then enter the circulation and go back to the muscle cell, where it can be broken down in glycolysis to make ATP and our original friend, pyruvate. This nifty little cycle is called the Cori cycle. Now, glutamate is a unique amino acid, and it's the only amino acid that doesn't have to transfer its nitrogen-containing amine group to another molecule. That's because glutamate undergoes oxidative deamination, a process that removes hydrogens and an amino group. So specifically, what happens is that glutamate travels to the liver mitochondria. This process takes place in the mitochondria because free ammonia in the cytoplasm can damage the cell. So within the relatively safe environment of the mitochondria, the enzyme glutamate dehydrogenase removes glutamate's amino group and adds an oxygen group from water, forming alpha-ketoglutarate. At the same time, glutamate's hydrogens get transferred to nicotinamide adenine dinucleotide, or NAD+, which forms NADH. The free ammonia in the mitochondria can then be converted into the less toxic molecule urea, in a process called the urea cycle. And remember that all of these reactions are reversible. So when the body needs amino acids, these reactions can go in full reverse, using the same enzymes to synthesize our original amino acid, alanine. Now, in addition to the nitrogen-containing amine group, the cell still has to metabolize the rest of the amino acid. Some amino acids, like alanine and glycine, serve as substrates to make new glucose, and we call those glucogenic amino acids. Other amino acids, like leucine and lysine, form alternative energy fuels called ketone bodies, and we call those ketogenic. And other amino acids, like valine and aspartate, can feed directly into the citric acid cycle and lead to the formation of energy as ATP. Some amino acids, like phenylalanine, can do it all, being glucogenic, ketogenic, and feeding into the citric acid cycle as well. Alright, as a quick recap, amino acids have a nitrogen-containing amine group that has to be safely metabolized to avoid toxicity. To do that, cells use transamination reactions with a keto acid like alpha-ketoglutarate to generate intermediates like pyruvate and glutamate. Glutamate can then be oxidatively deaminated in the liver mitochondria to get rid of ammonia in the urea cycle.
I really just wanted to watch that portion of the osmosis to give an example of the complexity of the amino acids and how they're used differently and what that would result with in terms of how they're used in their function. Let's keep going on the nucleotides with the AMP. So, the cyclic version of both AMP and GMP are important because they serve as intracellular second messengers. If we go down to this here, it shows that ATP serves as the main energy supply for cells, but it's also the precursor of the second messenger of the CAMP, right? So CAMP is formed when ATP undergoes a reaction that converts it into the ring structure. And that reaction is catalyzed by adenylyl cyclase and is often activated in response to the ligand receptor interaction on the plasma membrane. So CAMP then acts as an intracellular second messenger, turning on a signaling cascade through the activation of downstream kinases. So like CAMP, cyclic GMP also serves as an intracellular second messenger. And then finally, GTP plays a key role in signal transduction as its hydrolysis to guanosine diphosphate, which is GDP, then provides a timing switch for many membrane receptors. So, for this reason, many receptors in this category are referred to as G protein-coupled receptors. So kind of going off of that when we start talking about DNA and RNA and how they are made and how they function, okay? So we all know that DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And these are the biomolecules responsible for our genetic inheritance, DNA, and for ongoing cell function through our protein synthesis with RNA. We now know that the Human Genome Project showed us that we have approximately 3 billion DNA pairs of nucleotides arranged on 23 pairs of chromosomes for a total of 46 chromosomes within the nucleus of each cell. So DNA is produced by the enzyme DNA polymerase, which catalyzes the serial addition of deoxynucleotides to create the linear molecule. And that's the process of replication. RNA is produced by the enzyme of the RNA polymerase, which catalyzes the serial addition of the ribonucleotides in the process called transcription, and we'll talk about that more next chapter. So these large molecules have a high degree of similarity in chemical structure, but have significant differences as well. So bonds between the phosphate group of a nucleotide and the ribose sugar of an adjacent nucleotide together form the nucleic acid backbone. Okay? So the linear structure of DNA and RNA are similar because they have a polarity that is based on the ability of oxygen atoms on the 3' and 5' carbons of the deoxyribose or ribose sugars to the sites of the synthesis reaction. The synthesis of either DNA or RNA occurs in the 5 to 3 direction, etc. This here, the figure you see here, is figure 2.27, and this shows that a short segment of a double-stranded DNA illustrating hydrogen bonding between complementary bases adenine and thymine, which are joined by 2 hydrogen bonds, and guanine and cytosine, which are joined by 3 hydrogen bonds. This alignment is favored by the anti-parallel arrangement of the sugar phosphate backbones. The 5' end to the 3' end, or the 5' to the 3' orientation of one strand lines up against the 3' to 5' orientation of the complementary strands. That's a lot of words. What I want you to understand is that when the two sides of a double-stranded DNA line up, the hydrogen bond forms between the complementary bases. And that's important because that affects the process of transcription and replication and makes the DNA stable. Also remember, adenosine makes 2 hydrogen bonds with thymine, and cytosine makes 3 hydrogen bonds with guanine. That's kind of what they want us to know. So adenosine makes 2 hydrogen bonds with thymine, so A and T go together, and the C and the G go together. Also remember that DNA is the repository of our genetic information in our cells. It stores all the information necessary to produce proteins, and the information that is completely duplicated before a cell can divide by the process of mitosis. And that is what then passes on identical genetic information to our children, or our daughter cells. In addition, half of a parent's genome is passed on to offspring through the meiotic division of germ cells, producing egg and sperm. RNA has the major role of transporting the information stored in DNA to the ribosomes where the protein synthesis occurs. All right, hang in there. We're getting towards the end. Okay, next thread we're going to talk about is xenobiotics, the exposome, and its implications for health. So just as the field of metabolomics has been growing, there's an even more recent field of exposomics, which is a new field that shows promise in understanding the pathophysiology of disease. So this field investigates the exposome, or the study of all of the exposures of an individual and how those ultimately affect health. So it includes our environmental factors, our synthetic chemicals, our lifestyle choices, all of that put together to try to figure out why diseases happen to the way that they do. The concept of exposome is not easily defined. So it was initially envisioned as the exposure to the environment, factors, and their influence on disease, but then this definition has now expanded to include the totality of all synthetic chemicals or physical stressors, biologic agent, lifestyle choices, psychosocial factors, and X, Y, and Z. So the nature of the exposome is inherently dynamic as it's constantly changing based on the conditions a person is exposed to. So it's kind of a really vague, to me, there's such a exponential possibility of factors. I'm not sure how anybody could truly do that, but I think it's neat. That's wonderful that they're doing it. Xenobiotics are substances that are not naturally produced in the body. So those are things that any human could be exposed to through ingestion, inhalation, skin contact, and they can contribute to acute and chronic toxicities. So we've studied xenobiotics for a long time, such as with heavy metals. We know that large exposure to mercury is not good, lead, chromium, arsenic, etc. We also know the xenobiotics are what have helped us learn that we have microplastics as potential environmental toxins. So that's bad, right? We now know that microplastic particles have been detected in various human samples of blood or tissues of the colon. A recent study performed meta-regression analysis of toxicological impacts of the microplastic exposure in human cells, and that suggested adverse effects on various parameters such as cytotoxicity, our immune response, and oxidative stress. So xenobiotics is a new-ish area that is really hot, and a lot of studies are being done. The reason we want to study all of these is that, unlike genetic predisposition, the environmental exposures are modifiable, and we're trying to figure out then how can we help prevent certain disease progression. Okay. At the end of the chapter, they focus on phenylketonuria. And this example that they use is the PKU. So PKU is a disease that is inherited in an autosomal recessive fashion. So in autosomal recessive disorders, that means individuals who inherit 2 copies of the disease will confer gene development of the disease. So patients with PKU carry a mutation in the gene coding for the enzyme phenylalanine hydroxylase. So the enzyme is responsible for adding the hydroxyl group to phenylalanine, thereby converting it to tyrosine. So patients with PKU develop hyperphenylaninemia, there you go, which means they accumulate high toxic levels of the amino acid phenylalanine. So they also likely do not have enough tyrosine. So the metabolites that are downstream from this amino acid can be decreased. So we see hypopigmentation in childhood because they don't have enough tyrosine to produce melanin. And then they cause mousy odor of child's urine and when you can't convert phenylalanine into phenylpyruvate. So in the 60s, we started screening for PKU. It's now mandatory in all states. It's called, it falls under inborn errors of metabolism. Early intervention is key because we can improve outcomes. If an infant's diagnosed with PKU, they are maintained on a low protein, low phenylalanine diet. And so then they can avoid the many cognitive delays that can occur. So they're treated with strict dietary regimen. And it's now well established that adults have to follow the same dietary regimen to avoid behavioral disturbances and cognitive issues as well. Okay. At the end of the chapter, I would encourage you guys to read the key points. I think they're really good and handy to reflect on. So, what have we covered? Here's the key points. The body is composed of molecules that are made by chemical bonds between individual atoms. The properties of the atoms depend on their atomic weight and structure. The nucleus of an atom is made up of proteins and neutrons with a surrounding shell of electrons. Electrons are arranged in orbits, or their shells, and the atomic properties depend on the extent to which the outermost shell, which is the valence shell, is occupied by electrons. Biomolecules are formed when covalent bonds are made between atoms and molecules, building up from simple to very complex macromolecular structures. The 4 main elements in the human body that we need are carbon, hydrogen, oxygen, and nitrogen. Several other elements are found in lesser amounts. The elements are stabilized by either losing or gaining the electron, called ions, and several ions have those distinct biological roles. Covalent bonds in biomolecules can be characterized as nonpolar or polar. In nonpolar bonds, there's a relatively equal sharing of those electrons between the 2 bonding atoms, so they don't have a bunch of electronegativity. In the polar bonds, one atom exerts a greater attraction on the shared electron than the other. So because it's now unequal sharing, partial charges develop within the molecules such that one atom carries a partial negative charge and the other partial positive charge. In the biomolecules, structures containing only carbon and hydrogen are nonpolar, but structures containing hydrogen bonded to oxygen or nitrogen are often polar. Water is the most abundant molecule in the body, and it is the polar solvent of the extracellular and intracellular fluid. Substances that are charged are called ions, or polar are soluble in water and are referred to as hydrophilic. Hydrophilic is water-loving. Substances that are nonpolar are not soluble in water and are referred to as hydrophobic, or water-fearful. The biomolecules most found in the body consist of carbon atoms bonded to hydrogen, oxygen, nitrogen, phosphorus, sulfur, and some of the other lesser atoms. Carbohydrates consist of carbon, hydrogen, and oxygen. So they are polar molecules and include simple sugars, monosaccharides, that most have 5 to 6 carbons. The most abundant carbohydrate is D-glucose, and it's used as the primary energy source for many tissues, and it's stored in the form of glycogen. Okay, so that's our carbohydrates. Lipid molecules contain regions that are very hydrophobic, water-hating, that consist of carbons bonded to hydrogen. Our major body lipids are triglycerides, which represent a major body energy store, phospholipids, which makes up cell plasma membranes, and cholesterol, which contributes to plasma membrane structure and is the basis for synthesis of bile acids and steroid hormones. Amino acids, they contain carbon, hydrogen, oxygen, nitrogen, and sulfur. Individual amino acids have properties conferred by their unique side chains. Remember, amino acids are the building blocks of proteins and chains of amino acids linked by peptide bonds. Proteins are the most structurally diverse biomolecule in the body, and they serve innumerable structural and functional roles. Nucleotides contain carbon, hydrogen, oxygen, nitrogen, and phosphorus, and serve several roles in cell function, most notably the critical cell energy compound ATP. Nucleotides are the building blocks of the nucleic acids DNA and RNA. Chemicals entering the body from the environment and those ingested, inhaled, or applied topically are termed xenobiotics. These exogenous chemicals and their metabolic end products are potential sources of disease and continue to be studied. You've survived part 1 and part 2. I will attach these to your modules for handouts. Thank you.